Hi, this is Dr. Janine LeBlanc-Straceski from the Department of Biology at Merrimack College. This is the first in a series of lectures for a college-level course in genetics. The course is divided into 15 units, each of which will be presented in a few short videos. I'll cover topics including the structure of the genetic material, the central dogma, the genetics of microbes, Mendelian genetics, personalized medicine, and a whole lot more. The framework of this course is based on the Robert Burker text Concepts of Genetics, with a lot of additional material from various sources. Credits for this material can be found on the PowerPoint files in my website. Let's get started with the first unit, the molecules of heredity, structure, and function. The first lecture for Unit 1 discusses the experiments that led up to the discovery that DNA was the genetic material. The learning goals for this unit are to understand the major experiments that led to the discovery of DNA as the genetic material, what those experimental methods were, and who the investigators are that were involved. It's also to understand how the properties of the DNA molecule itself led to those findings. Genetic material must be able to store information, a lot of information, enough information to make an entire organism. It has to be able to be transmitted, passed on from parent to offspring. It has to be able to be replicated, also so that it can be passed on from parent to offspring. And it also must be able to account for variation. We know that mutations happen, and we know that a population of an individual species has a fair amount of variation in it. The genetic material has to be faithfully replicated, but it also has to be able to account for certain amounts of variations within a population. So, keeping in mind all of these important criteria, let's find out how DNA was discovered to be the genetic material. Back at the early 1900s, the debate raged. What was the genetic material? There are four major groups of molecules in a cell: lipids, carbohydrates, proteins, and nucleic acids. Which of those different molecules was capable of encoding the genetic material? Well, for the most part, scientists were convinced initially that proteins contain enough complexity to be able to encode all the information required to make an organism. But DNA was lurking in the background. There was some experimental evidence that indicated that maybe it might just be DNA. The experiments by Fredrick Griffith, Avery McCarty and MacLeod, and Hershey and Chase really supported the hypothesis that indeed it was DNA that was the genetic material. So let's look at Fredrick Griffith's experiments first. Fred studied a particular type of bacteria called Streptococcus pneumoniae, or pneumococcus. Streptococcus pneumoniae, or pneumococcus, came in two different major varieties. Both of these pictures here indicate the two different strains of pneumococcus: a rough strain and a smooth strain. The rough strain is called rough because the surface of the bacteria is actually exposed. The smooth strain is called that because in the middle of this glob is actually the same pneumococcus cell and coated with a very sticky, gooey mucus that has been secreted by the cell. Now, these types of cells were called smooth, these types were called rough. Now, the difference between these two as far as virulence is concerned is very interesting. The rough bacteria would not make you very sick. If you were infected by some of this strain of pneumococcus, your immune system would have easy access to the surface of this bacterial cell and be able to clear it from the bloodstream. so that even though you might be sick for a little while, your immune system could handle it just fine, and you would recover quite well from an infection by this rough strain of pneumococcus. However, if you are unfortunate enough to get infected with the smooth variety, this heavy mucopolysaccharide coat around all the bacteria kept the immunoglobulins from your immune system from being able to attach to and destroy the bacteria cells that were encased in this polysaccharide coat. Okay. Griffiths was familiar with these two different strains of bacteria, and with an experimental approach called heat killing. Let's look at his experiment. If you take a mouse and inject into it some live smooth bacteria, after several days, the mouse succumbed to the infection. If you take some blood samples from this mouse and culture the bacteria from it, you were able to recover some S bacteria, some smooth bacteria from this culture, from this mouse. Okay? So you were able to recover some live S bacteria. You put live S bacteria in, you got some live S bacteria out again. However, if you use the R strain instead, the mouse was able to mount an immune response, clear the infection. The mouse survived, but you never recovered any R bacteria from the mouse's bloodstream because the immune system of the mouse killed it all. So no living bacteria were able to be cultured from this mouse. Okay, so here comes the interesting part. If you heat kill, in other words, you take some S bacteria and you heat it up, boil it essentially, and you inject those into a mouse, what happens? Now you have some dead S bacteria. Okay? The mouse can handle this dead bacteria no problem. The S bacteria does not multiply inside the mouse. What's left over, the mouse can easily clear. Its immune system can easily clear. And if you try and culture some bacteria from the mouse's bloodstream, nothing. There's nothing there. Okay. The mouse survived, and you are not able to extract any living bacteria from the mouse. Okay, so how do you know if there's some genetic material in the cell? This is the next step is Griffith's very interesting concept. He took some living R bacteria, which if you gave it to a mouse, the mouse is perfectly fine. And he took some heat-killed S bacteria, which also if you give it to the mouse, it's perfectly fine by itself. And mixed them together and injected it into the mouse. And what happens to the mouse? The mouse dies. Okay? It dies from a pneumococcal infection. And if you take some of the blood and culture it, you end up with some living S-type bacteria. How is that possible? Well, what's happened here? This is not a Frankenstein experiment. We didn't resurrect the dead S bacteria. What happened was some kind of molecule that was present in the S bacteria that made it an S-type bacteria was transferred to the live R bacteria. And these bacteria cells were transformed from rough to smooth. Okay, something turned these rough bacteria into a smooth type bacteria that caused the infection, that caused the mouse to die. Okay, so Griffiths did these experiments in the 1920s, and people were interested in it, but we still didn't know exactly what the genetic material was. We knew it was a molecule. That was a big step forward. We knew there was some kind of chemical basis for heredity. When was DNA proved to be the chemical basis of heredity? Well, it took until 1944 when three scientists, very dapper scientists here, Oswald Avery, Colin MacLeod, and Macklin McCarty, got together and tried their own version of Griffith's experiment with an important twist. So they carried out their experiments in the 1940s. Instead of just mixing a dead and alive bacteria, they took some of the rough bacteria and mixed it with specific extracts of the S bacteria. They fractionated the S bacteria into lipids, carbohydrates, proteins, and nucleic acids, both DNA and RNA. It was hard to separate at that point DNA from RNA. So they had, in their preparation, they had both nucleic acids, DNA and RNA. Oh, and they also took some time out to join the band. Okay, so here's their experiment. They took some type R cells, and they mixed the cells with a small percentage of R cells, mixed the R cells, and looking for a small percentage of them to be transformed into S cells. They were able to eliminate the R cells with antibodies and centrifugation. That's a very key step in this process. And they plated the remaining bacteria on Petri dishes. Okay, so here's your R cells. If you add an antibody against the R cells and plate it, what happens? Nothing grows because they've all been cleared. If you mix the R cells with some S cell DNA and let it grow for a little while, and then use your antibodies to clear all the R cells away, you do, but you end up with some S cells, some smooth bacteria cells, growing on your Petri dish. So here, your R cells are transformed into S cells. Okay. Now, the next thing they did was to try and prove that in their extract, it was the DNA that was doing the transformation. There were a lot of skeptics. They said, oh, no, it's not the DNA. Instead, you've carried some contaminating molecules along in your extract, and it's really the protein that's causing this transformation to happen. So, Avery McCarty and MacLeod treated their extract with DNase. That would destroy all the DNA in their preparation. And when they completed this experiment, they got no transformation, indicating that indeed it was the DNA in this prep that was transferring the genetic information from the S cell extract into the R cells and transforming the R cells into S cells. If they treated their extract with RNase instead, they got transformed. So it wasn't any RNA in this, because if you eliminated the RNA, you still got transformation. And also, if you treat with proteases, you were still able to transform. So if you eliminate the protein, the DNA that's still remaining in their extract was able to transform the R cells into S cells, and they were able to culture their S cells. So that was a very good demonstration that DNA was probably the genetic material that was able to transform R cells into S cells. Okay, one more experiment we're going to talk about, a very important experiment by two investigators called Alfred Hershey and Martha Chase. In 1952, they used a newfangled approach. They used radioactive isotopes that would label specifically the proteins and the nucleic acids with tags that could be traced easily by their radioactivity. They studied the bacteriophage T2, which is composed of only two macromolecules. So there's no lipids, there's no carbohydrates, there's only DNA and protein. And here's a model, courtesy of the C phages class, where you can see the protein coat and the DNA on the inside, right in here. This is your DNA in here, and in here. Okay. Here's a model. This is an actual electron micrograph of a T2 bacteriophage, and here is a schematic diagram showing all the different parts of the bacteriophage, the capsid, the protein capsid, which encloses the DNA molecule inside here. Okay, so just to refresh your memory, if you don't know or haven't forgotten, when a virus infects a bacteria cell and injects its genetic material inside the bacteria cell, and that genetic material then directs the production of all the different parts of the new phage particles, which eventually assemble and then cause the bacterial host to burst open and release all the new little baby phages into the environment. Okay, so this is another schematic diagram showing that if you label the DNA inside the capsid head with radioactive phosphorus, P32, or the protein coat with radioactive sulfur, S35, you can follow these two different macromolecules, either the protein coats or the DNA that was inside, by measuring the different emissions of the two different radioactive isotopes. And this is their experiment. The first thing they did was to prepare E. coli cells, that's the host, the bacterial host, with by growing them in either radioactive sulfur or radioactive P32. Okay, so here's your E. coli. You're growing them with some radioactive sulfur or some radioactive P32. And you're going to grow up a whole bunch of radioactive bacteriophage or viruses that contain their S35 either on the protein coat on the outside or the DNA on the inside of the virus particle. Okay, so after you recover these viruses, you are then going to again infect new cultures of bacteria. And you're going to infect some new cultures of bacteria. You're going to add the S35 T2 bacteriophage labeled to one flask, and your viruses labeled with P32 to the other flask. And you are going to allow the infection to occur briefly, just long enough so that your phage particles have enough time to sit down on the surface of the bacteria and inject their DNA into the bacteria, but not long enough for them to replicate and burst open the bacteria cell. Okay, so you allow the infection to occur, and after a short period of time, you're going to agitate the solutions in a blender. You put them, the S35 labeled bacteriophage into one blender, your P32 labeled bacteriophage and bacteria into another blender, a few quick pulses, and you can knock the phage particles off of the bacteria. And then spin it in a centrifuge, and you end up with your bacteria in the pellet and your phage in the supernatant. Okay? And you count the radioactivity in either the bacteria or the phage to determine whether or not you've got your DNA, your genetic material has ended up in the bacteria, or if it's remaining behind in the protein coats in the supernatant. So here's the test tube with the bacteria at the bottom and the supernatant up here with the empty coats. Okay, the empty bacteriophage coats are up here, and the pellets contain the bacteria cells with whatever the genetic material is in it. And you use the Geiger counter. This is something we used to use in the lab all the time, and it's not used very much anymore. But this Geiger counter will be able to detect radioactivity at different parts of the test tube. So you're going to count the amount of radioisotope in the supernatant with the Geiger counter and compare it with the starting amount. So what happens if you've got S35 labeled bacteriophage in this test tube? Where do you think the radioactivity is going to be? Do you think it's going to be down here with the bacteria, or is it going to remain up here in the supernatant? And the same question for the P32. Where is it going to be? So this is the data. This graph shows the amount of radioactivity just in the supernatant. Okay, so it's not looking at the pellet at all on this graph. We're just looking at the amount of radioactivity in the supernatant. Here's the supernatant. Okay, so when you label the bacteriophage with S35, which labels proteins, you find most of the radioactivity, 80% of it, at the end of this experiment, in the supernatant. Okay? But when you label it with P32, you find very little of it in the supernatant here, and most of it is in the pellet. So that means that when you label your bacteriophage with P32 and they inject their genetic material into the bacteria, that most of it ends up in the pellet, which means that the DNA is the genetic material. Okay, so most of the S35 was found in the supernatant and a small amount of the P32 in the supernatant, but most remained with the cell. So in summary, the identification of DNA as the genetic material is based on 3 key experiments. Griffiths showed that there was a molecule that was associated with heredity. Avery McCarty and MacLeod showed that it was DNA, and so did Hershey and Chase. That the genetic material is stable and molecular in nature, it's transmissible outside of a cell, it's destroyed by DNase, but not by protease or RNase, and then it's identifiable as DNA by its phosphate content and lack of sulfur.